Now, without any further ado, we will start our first session. This session will be moderated by Dr. Safarina Malik from the Mokhtariyad Institute for Nanotechnology, Indonesia. Dr. Safarina Malik, the time is yours.
Thank you, Priska. Without further ado, I would like to invite our first speaker, Professor Gloria Dominguez-Bello, to give her first presentation regarding the history of the Microbiota Vault. Gloria, the time is yours.
Thank you so much.
You have 15 minutes. You have 15 minutes, okay?
No problem. Can you see my screen?
Clearly, yes, very clear.
Okay. Well, I'd like to start by giving a short history of the Microbiota Vault, a global non-profit initiative to conserve long-term health for humanity. And so these are images of traditional peoples that live in the jungles of South America. This in particular is peoples from Venezuela, my country of birth, who live in very remote, isolated places, and that I have been studying. I've been studying healthy people that live in remote places away from market economies and medical exposure. And what we have seen is, as we move from very traditional lifestyle to cities, the diversity of the microbiota in the gut decreases. And this is depicted on this left side graph, where you can see Amerindians from South America, very isolated or less isolated, the Guahibo is another more assimilated group, or people in Malawi with similar, but then another step down in the US. Numerous people have shown these associations between lower diversity and higher urban lifestyle. And at the same time, there is a correlation with an increase in metabolic and immune diseases. And this is one of the biggest challenges that medicine has today. We seem to be replacing infectious diseases by chronic diseases. And this is the fate when countries integrate into market economies. So the challenge is there an association between the microbiome and these diseases? And a lot of epidemiological work shows that there is, and a lot of increasing evidence from mouse studies show causation. It's not surprising, because the microbiome has a lot of relevance for health, as we all know, in metabolism, immunity, in the nervous system, everywhere, basically because these microbes that live on our epithelia are signaling, our host cells are signaling the immune system and are signaling the brain, and it's affecting physiology in general. There are almost 200 times more genes that live on the microbiome and are signaling us than human genes are. So they are an important part of genetic expression regulation for us. When we perturb the balance with antimicrobials, impairing transmission in early life, etc., these functions are altered. So what we reasoned was, okay, if as we move from jungles and savannas into rural, into urban, if we are increasing modern diseases in correlation with urbanization, as we degrade the microbiome diversity, can we restore? Can we give back the microbes, and will that revert the trend in modern diseases? This is what we propose. And for this, what we need is to preserve the microbes, because if we don't, by the time we know what is the importance of specific microbes, we will have lost the microbial diversity. I'm originally from Venezuela, and my collection that I had there and that I moved has been subject to a lot of threats, from political to hurricanes, because I moved to Puerto Rico and then to New York. And of course, our collections are subject, even in the most secure countries, they could be subject to somebody unplugging or an electric failure in the freezer. So it's a big responsibility to have just your research collection without more security and being more sure that the collection can survive an accident, for example, in your lab or in a local collection. So putting together all these, what I was seeing from my work, the disappearance of diversity, and what I was going through, in Venezuela with the unrest, and then I moved to Puerto Rico where there are hurricanes every year. And when I moved to New York, Hurricane Sandy was probably the one that was closest to completely destroy the collection. I learned from the Svalbard Global Seed Vault. And this is an effort that Gary Fowler moved forward for many years until he achieved a collection of seeds from everywhere, from local collections everywhere in the world, that can deposit there, and is a storage repository in the permafrost of the ice in one of the Svalbard archipelago islands in Norway. So I connected with them, and this is kind of a timeline. Venezuelan revolution, a lot of unrest, and then I moved to Puerto Rico, hurricanes. I moved to New York, another hurricane, while we were learning about disappearing microbes. And I learned about the seed vault. And then in 2018, I met Manuel Fankhauser, who is a program officer in the Sea Rate Foundation. He was a recent program officer, and he was screening for projects to fund the foundation. The Sea Rate Foundation is from Switzerland. And Manuel and I spoke, and I told him about the idea of the Microbiota Vault. And it was really Manuel who materialized the whole thing, because he's an action person. He executes, and he was able to, help materialize and put together plan the feasibility study, which was beautifully performed in Switzerland. And now the person who worked on the feasibility study is Dominic Steiger, and he's an officer of the Microbiota Vault. We have started at the end of 2020 the pilot phase of the Microbiota Vault, and also in Switzerland. And in this pilot phase, very prominent and well-known scientists of the microbiome, Pascal Bonash, Adrian Egli, and Nick Bokulish, are putting together the studies to understand how to preserve the different microbes, but also receive actual deposits in this pilot phase. The deposits are currently in a laboratory of Adrian Egli, but the idea is to have our own vault. And the pilot phase, because it depends on the money we could raise, we raised money that was purposely for human microbiome and for fermented foods. The Rockefeller Foundation is interested in studying and preserving microbes from fermented foods in the world. And the whole effort, in the whole effort, we want to open and also include in the future environmental collections. So we want to preserve the microbial communities that are important for the planet and therefore for humankind. We put together a remarkable team of very prominent researchers. There are 2 Nobel Prizes in the board. Everybody that has read on microbiome have read of many of these scientists here. And the idea is to be a foundation that offers a backup repository of microbial specimens to local collections. So the local collections are really the feet of the whole system, the columns that sustain the system. And then we, through GloMiNe and other efforts, we give capacity building. We try to help the collections that have not enough means, especially considering that the hotspots of biodiversity are in developing countries, precisely because they have not fully industrialized yet. So before they do and they lose the microbial diversity, we need to preserve. And we have the goals and the challenges are of different natures: technical, legal, organizational, social and ethical and economical. Here we see Dominique Steiger, the creator of the feasibility study and now program officer in the Microbiota Vault. And on the right, you can see the support of all the institutions that we have received, and thanks to which we are now in the middle of this pilot study. These are the 3 scientists I mentioned before. All 3 are working in Switzerland. And Tore Midve has been moving efforts ahead in Norway. And, you know, with the very unstable world we have today, we are considering also exploring to have some redundancy and have more than one site. So the idea, as I said before, this core of the whole effort are local collections. Local collections can request to us to preserve a copy, a backup, and as we would do it for free as a service. And we will also, to the possibilities of our economic possibilities, ask them if they want the metagenomic sequencing to be done also for free, with the condition that the sequences are open, are deposited in accessible databases to the world, for the world to research. This is a very important point because in many developing countries where there is not enough money for a wet lab, if you train the people to do the analysis of data, the amount of data that is being published in every paper, and it will be published with this effort, is enormous. So just with the data that is available in the world, there's a lot of research that can be done and a lot of analysis that needs to be done. So
Gloria, you have 1 minute.
Yeah, so just to wrap up, the effort includes not only storage and metagenomic data for available to the world, but also education, of which this GloMiNe is an important activity that we plan to do every year. I love this. I took this photograph in New York, trying to, you know, put the ball in the basket. Be together is not the same. We need a whole global village to achieve this purpose. And with that, I'm glad to answer questions.
Thank you, Gloria. For the audience, we will have the question and answer at the end of the session.
Perfect.
Thank you, Gloria. Now we will continue to our next speaker, our second speaker,